A warm good morning to one and all. Today we are coming to a new aspect of bioinformatics, an application aspect of bioinformatics. Now, the most important or the most hot topic of today is to find a drug or a vaccine against COVID 19. And we all know that it is found to be a very tedious task. And COVID 19 had emerged in November 2019. And even now, we are in fact reached a stage at which we have a clinical trials of the particular vaccine. So fast is the emergence of the vaccine research as well as the attempts which have been carried out to find a vaccine against COVID 19. The usual purpose of drug designing normally involves the taking each and every plant or medicinal medicine which is available for us, trying to give that to a person who has been infected and to check whether the symptoms are gone or not. Each and once you get a positive result for that, what happens, they will do first, it will be done on mice, then it will be done on humans, and once negative effects are not being found for a drug, what do? We will chemically synthesize that particular drug at a later stage. And the whole process of this drug designing usually takes about 2.5 to 3 years minimum for a pharmaceutical company to reach to a conclusion, and large amount of money is being invested in such drug discovery process. In the wake of necessity, when we need an immediate result, at the same time when we want to spare the money which we have also for the extensive research which can be done for in the case of drug discovery, there are some options which come across us which will help us to lessen the time which is being needed for a drug discovery. And bioinformatics is one such of a technique. Now you might think, how does this work? In the process of drug discovery or the computer aided drug discovery, in which you use bioinformatic tools to discover the drugs, what you do is you take a particular target. Now, if you consider if a particular enzyme is very much critical for a microbe or for its growth, we will take a particular that enzyme as a target to which the drug has to be directed. And that drug, the PDB structure as well as the protein structure of it, you know what is that? Okay, the three-dimensional PDB structure based structure of that particular drug is being created using bioinformatics, and at the same time various other target molecules. Okay, now you have different drug molecules which we have. They are also being created, and in the computer, okay, we will look three-dimensionally. Will they interact with the particular protein? And will they inhibit? Okay, you might think, what is the point in just interacting? Always the efficiency of a particular enzyme, it depends upon the binding of a substrate to its active site. So if a critical enzyme which is involved in disease has an active enzyme, it has an active site, okay, and if that active site is being blocked using something so that it cannot carry out the reaction which it usually carries out, what will happen? We would be able to control the particular disease. The same principle here is being used in bioinformatics, in which what happens, you will take a particular target enzyme, and that target enzyme's active site the whole protein is created using the help of the computers. That is, you will prepare a 3D structure of that, and then at the same time you will create 3D structures of different drugs or various molecules which you suspect to have an interaction with the active site of the particular enzyme. And so when you have different drugs, you will screen all these drugs which will interact with the what? With the active site of that particular enzyme. Okay. And if and you know there is a binding energy with which active protein will interact with each other. So if these if the molecule of our concern is having a very high affinity for the active site of the enzyme, what happens? It will bind at a at a say. And so the purpose is, instead of letting the real substrate to come and bind to the active site, we will give this drug molecule so that if it interacts with the active site of the critical enzyme, it will prevent the progression of the particular disease. So using computers, we will first create both the active enzyme as well as the targeted prospective drug molecules which we consider in our study, and then we will try to see if they are interacting with each other. Okay, so we are just preventing the real substrate of the enzyme to interact with it. So the prevention of the enzyme activity is one way by which you can prevent the progression of a particular disease. Now, at the same time, you should also know something or term over here called the binding energy. For every interaction between molecules, there is a binding energy. And if it interacts with the least binding energy, it means that it is having a higher affinity. That is, less amount of energy is being needed for its binding. So the least of binding energy you need for a target molecule to interact with a protein, what is it? It means that it is having a high affinity, or it can be even in a normal system with less amount of energy it will bind. In all the biological systems, it makes a plan that you should get the molecules which will interact with least binding energy. We always try to have things which will have limited amount of energy, or which needs limited amount of energy within our body. Now, in the case of drug designing, we are using a technique called docking or computer aided drug designing, where in which this interaction between the drug molecule as well as the targeted molecule or the prospective drug molecules are being created using the computer. Now you might think, why do I have to spend so much of research into it? Now, if you have lots of drug targets, then what you will do? You will make all the 3D structures of all these drugs, and then after that you will make them interact with a particular enzyme. And among the hundreds and millions of drugs, drug target molecules, what do you see? The ones which are having the least binding energy will be first selected. And then you will see, okay, fine. These are the ones which could be the prospective drug target molecules against this particular enzyme. So in the process of drug discovery, you take first and foremost, what do? You find out the target, a biological target, which can prevent its replication or prevent its growth in a host. Okay, so that is one thing that we need to find out, and then we create the PDB structure of that using bioinformatic tools, and at the same time you will create a list of all the prospective drug molecules which are there, and we will see whether they are interacting with the active site of binding with the active site of this particular protein using computational tools. So hope you got an introduction to what we're going to do with bioinformatics in the process of drug discovery. We'll be continuing in the next session on more details with figures and all that on the process of drug discovery and what is the need of it in the coming sessions. Thank you for now.